Our laboratory is studying the mechanisms that control chromosome integrity and therefore that prevent genome instability, which is a problem in all cancer cells because it has a causative role in tumor formation and development, as well as the relapse of two chemotherapeutic agents. For many years, we have been studying the mechanisms and the regulatory pathways which are calling out when cells is exposed to genotoxic insults, the so-called DNA damage response. And in particular, we have been studying the mechanisms that couple chromosome replication with DNA repair and recombination. Basically, what we did was to induce DNA damage in S phase and studying all those processes that maintain the conformation of replication forks, preventing aberrant transitions. More recently, we are interested in trying to understand which is the essential role of the so-called DNA damage response kinases, in particular ATR, which is a PI3 kinase, and particularly to try to understand which are the essential and physiological function of this kinase under amplitude conditions. So it turns out that the key role of these proteins is to coordinate the clash between transcription and replication, which is a big challenge for the cell. Lots of things may go wrong. So basically the way I see it is like two trains that clash head-on the same track. And we believe that this is a huge problem for oncogenes because oncogene induce the so-called replication stress, which is likely due to a scheduled transcription event in S phase. So the coordination of this event is crucial for the cell. And when something goes wrong, you have a tremendous accumulation of genome damage. A question that we are trying to address more recently in these days is which are the sensing mechanisms that allow the cells to be able to monitor and to sense the clash between replication fork and transcription units. Turns out that recent data obtained in our laboratory really suggests that the cell is able to sense the mechanical forces which are generated by chromosome dynamics at the nuclear envelope, not only during chromosome replication, but also during condensation. And ATR plays a huge role here because it acts as a mechanosensor. In fact, we really open a new field here because this kind of process really couples chromosome dynamics with nuclear envelope metabolism and mechanotransduction pathways acting inside and outside the nucleus. There's another line of research in the lab which is more involved in trying to understand which are the connections between the metabolic processes and DNA damage response and chromosome integrity. And in particular, we are studying the links between the main PI3 kinases, mTOR, ATI, and ATM.